Again, good morning, and my special thanks to the Pro Vice Chancellor, Professor Ajay Kumar, Dr. Ganga Prasad, Dr. Deshmukh, Professor Jayakumaran Nair, which we have a collaboration, Professor Sudhagaran, and my special thanks to Professor G.M. Nair. I assume that he will be there because he is the one who introduced me to Kerala University and Inter-University Center for Gene Technology. And because of that, first time, probably 5-6 years back, he called me to give a talk there. And subsequent to that, I became advisor to Rajiv Gandhi Institute, then Sri Chitra Institute, Inter-University Biomedical Center, and now Kerala Life Sciences Park. Thank you, Professor K.M. Nair. Also, we initiated a pair program with him, KSCST, our company, as well as another company where I am also an independent director, and we are starting multiple other pair programs. This is all because of Professor G.M. Nair. Thank you, Professor G.M. Nair. Now, I'm given about 40 minutes and I have about 36 slides. Hopefully I will be able to finish. And this is the real work which we have developed a product uniquely in India, indigenously, everything. And the first time a molecule for Wet-AMD as well as diabetic retinopathy. What I'm going to talk is on discovery and development. So the discovery part I will cover briefly, and how the molecule is developed, and using synthetic biology. Synthetic biology is like quite different from the other biotechnological process what is used, and development of an anti-VEGF antigen binding fragment. So antigen binding fragment, part of the antibody, that's what we have developed, which binds to VEGF, vascular endothelial growth factor. Two diseases we have developed. One is Wet-AMD, or age-related macular dystrophy, and diabetic retinopathy. So one of the molecule I will tell you in details, and I will also briefly tell about various startup companies, very successful companies. So Cygenome is our incubation company based at Cochin. Then Saksin Life Sciences is the first molecule we have developed in India. And let me tell you, this type of work is not taken over by any pharmaceutical company because it's extremely risky. A lot of money is required. The same way, Theragen Biologics, which is involved in the development of diabetic retinopathy molecule. We also have Theragen Lifesciences involved in the development of AND, abbreviated new drug application. And one of the most successful company is Maggenome, which was a technology on nanoparticles which I developed a few years back with few PhD programs. Now we have translated, and especially we have developed the entire COVID products, which is viral transfer medium, molecular transfer medium, and extraction of RNA. That's one of the key steps in the determination of the viral detection or for the RT-PCR. So that also we have developed. And another very successful company what we developed is Medgenome. Only I'm an advisor. The other companies I take care. Medgenome is a Bangalore-based company, mainly involved in next generation sequencing, and we have a lot of research projects. And we have another company called Theragen Molecular Innovation, which is initiated now. Then in Cochin, we have Cygenome Life Sciences Park. We also have a program supported by Kerala State Industrial Development Corporation called Biotech and Biopharma Skill Enhancement Program. But because of COVID or pandemic, we had to stop, and hopefully we'll be again starting that. So I'll be just briefly going through about the introductory part, what exactly is Wet-AMD or diabetic retinopathy, and briefly I will also tell about the company overview. Then in detail, I'll discuss about the development of one of the molecules, FAP201, which is an anti-VEGF for macular degeneration. And another molecule what we developed is diabetic retinopathy molecule, but I will just briefly tell only. And what are the regulatory approvals needed? So all these things I'll probably will be able to tell very briefly. Now, since this lecture is on biopharmaceuticals, you should know what exactly is biopharmaceuticals. You might be amazed to know about the drug discovery program. Like, hardly in India has not developed any single novel chemical entity or biologics. What we are doing is either generic molecule or biosimilars. Only 7 countries in the world could produce drugs, novel drugs. And mainly it was started about 100 or 70, 80 years back rational drug discovery, and we have only 400 drugs, and probably we have about 35,000 various diseases are there. Now this includes, about huge number for neurological disease, a large number for psychiatric disease, a large number for cardiovascular disease. But many of these diseases do not have drugs, which if you go and analyze, you will be able to understand. And the last 80 years, we have been continuously developing small molecules, which is otherwise called NCE. And what Indian companies are developing, the generic version of this, that means they don't have to do the discovery part, development part. Once it is developed, the rest will be taken care of. But now it is going to be quite tough because during the patent period, you are not allowed to do that one unless you develop yourself. In the last 20 years, biologics molecules have started coming up because it has become very difficult to develop NCEs, new chemical entities, or small molecules. That's why the large molecules, protein molecules like insulin, interferon alpha, interferon gamma, erythropoietin, these type of molecules have come. But if you see last 10 years, the major developments are all antibodies. Antibodies against surface antigen, which stop the transduction process. So those type of molecules are there. And what I will be talking is about the development of a novel biotechnology based or synthetic biology based Fab molecule, fraction antigen binding molecule against two different diseases. And if you see, there is a big difference between the chemical drugs or generic molecule or NCEs and biologics and biosimilars. Biologics is something called novel molecules. That's what we are doing. Biosimilars is that, existing those biologics molecule to repeat that one. Even that itself is a very complex process. Major difference is that, chemical drugs have very low molecular weight in the range of 200 to 600. On the other hand, biologics molecule normally about few hundred amino acids. Few molecules are peptide molecules. For example, molecules like leuprolide. So otherwise, it is normally biologically originated and probably in the range of about few thousand molecular weight. So that's the major difference. Small molecular NCs are very simple structure, and the 3 dimension of a large molecule is very complex. So extremely complex structure for biologics molecule. Now, on the other hand, NCs are normally chemically synthesized, normally 99%. On the other hand, the biologics molecules are produced by living cells. Now there is a process called synthetic biology, and that's what we are doing, and that's what we have developed this molecule. And always small molecules have a standardized manufacturing process. On the other hand, biologics have a highly specialized manufacturing process, which I'll be briefly mentioning. Normally, small molecules, exactly the ditto one will be able to, otherwise nobody will approve that, 100% equality should be there. But it is very difficult to develop. Even you can have the sequence, amino acid sequence for biologics, but conformation can slightly change. So those, the minor changes, it is probably sometime the regulatory people will not give approval. So it is a very complex process to develop biologics molecule. Normally NCs are very stable, but on the other hand, large molecules sensitive to storage and handling. Most of the small molecules are non-immunogenic. On the other hand, large molecules are non-immunogenic. Sorry. Yeah, this is all about the difference between NCEs and biopharmaceuticals. And what I am going to talk is mainly on the biopharmaceuticals. Here you can see a molecule move around. This is actually the antibody what we have developed. This is the 3-dimensional conformation of the molecule. This is really the molecule what we have developed is for the anti-VEGF molecule, which is for Wet-AMD. Now, this is what we need to develop, this molecule. If you see, this particular molecule has got about 500 plus amino acid, 48 kDa. And if you see an antibody, which is normally about 148 to 150 kDa, about 1500 amino acids will be there. And the Y-shaped one. If you see the Y-shaped one, the down part is the constant region. The other parts are the various variable region which constitutes the CDR, complementarity determining region. So that is what we have to focus on. What is the complementarity determining region? How we have developed using synthetic biology technique. So this particular antigen binding fragment for treating wet AMD, I'll be talking to you in detail. Now, what about the disease? As I mentioned, two diseases I'll be focusing. One is macular degeneration. So what is this disease? It is actually degeneration of the retina, neuronal cells. And how this is happening because of the genetic abnormality, and people who are above 45, 50, 55, normally above 60 it is happening. Macular degeneration, there are two types of degenerations are there, AMD or dry AMD. Normally a very large number of people have got dry AMD, but the problem is that 15% of the dry AMD people will be converted to wet AMD. Now, once it is converted into wet AMD, what will happen is that, neovascularization takes place. What is neovascularization? Vasculature or blood vessel in the eye, especially retina. Now, if you see the retina, the key vision area is called macula and fovea. Macula is in the central part of retina, and inside that the fovea, which is giving the key vision. Now, if something happens to that by degeneration or any other process, then you will lose your vision. That's a major problem. Now, in this case, what is happening? This is the normal macula, and this is the wet macular degeneration. So I mentioned about neovascularization or lot of vasculature. Blood vessel will come. If this blood vessel increases at a particular stage, it breaks, and the blood will ooze out, and the blood will come in front of the retinal, macula, and fovea region. Then you will lose the vision. Now, this is not a small number. A very large number of people can't see this one. Exactly the same way, diabetic retinopathy. And you know that India is called the capital of diabetes. A large number of people, in Tamil Nadu, I think about 12 to 15% of the people have got diabetes. And if it is above 50, 55, about 45 to 50% have got diabetes. Now, the problem is that diabetes is one, and because of diabetes, the glycosylation takes place, and many protein can get, abnormality. And one of the major problem is that the retinal damage or the glycosylation of the retina, by which again neovascularization takes place. And once neovascularization takes place, exactly like macular degeneration, you will lose the vision. It's a very large number of people. And probably in India has got about 5 million people are expected to have wet AMD. And diabetic retinopathy probably about 15 million. Both are very serious disease, and you won't be able to see. But what is the major problem? There is only few drugs. One drug only is developed in the US and extremely expensive. This is the background that we wanted to develop. Can we develop in India from this type of molecule? And we were not telling about the biosimilar or repeating the same drug. We wanted to develop a totally new drug which is going to be inexpensive. Right now, a single injection of these drugs costs $2,000 per injection. Like that, you have to give about 10 to 12 injections, which will cost probably about $20,000 to $25,000, which is not affordable even for the rich class. I'll just briefly tell about our company overview before further going. It's a Chennai-based company, and we are only developing patented technology, worldwide patented products, because otherwise, no one will fund us or, this VCs. If VC has to fund, it has to be like, patented products, and they do not mind having a risk in developing. But then what they want is that, if we invest, probably the product has to be new, novel, and there should be a very good return of their investment. That's the main thing. And what right now, these two companies are focusing on anti-VEGF based products for multiple ocular and certain oncology applications. So right now I'll be briefly talking about these two molecules, but many other things we are in the process of developing. We have, as every company, what the specific mission is there, and we wanted to make a differentiation in whatever we are making. We have a highly experienced team. That was one of the toughest things for us, getting the team, getting ready to develop this one, because it is very, very difficult to get experienced people. So we have to train them. And we have a very good track record of developing many products. Yeah, these are the different places. You can see the first one is the Donnelly Center in Toronto. So we have a collaboration with them, and the fast display technology, where the synthetic biology we are doing that. Now we have brought this to Cochin. This is QB3, which is in California. We have a laboratory there. This is one of the labs in Tysell Park, which we have the first molecule, what I am telling is developed there. The place I am sitting right now, we have a 60,000 square feet facility, which is not there. And this is another facility in Foster City. Part of the facility we are also using there. Now, I'll just briefly tell about anti-VEGF fab and its applications. Wet-AMD indication. As I mentioned, it is a patent protected. We have multiple countries issued the patents, and complete the product development attributes we have completed, and it is entering into a phase 1 trial. That also I'll briefly tell. What is the specialty? Now, if you are developing a molecule, whether it is NC or a novel molecule, if there is some improvement in the existing molecule, it is very difficult to commercialize. It is called superior molecule. Otherwise, the molecule has to be non-inferior. If suppose an inferior molecule you are developing than the existing one, there is very little chance that regulatory people will approve. So it has to be either non-inferior, but then you definitely have to give some additional application. But if the molecule is superior, and if you are doing everything properly, you will be able to get the approval. The other things are that, minimal toxicity. One of the problem with the anti-VEGF molecules are that it has to be injected into the eye. Many molecules have got problem in the toxicity or problem in the efficacy. So we will have to overcome all those things. And another thing is that this is a biologics molecule or a protein molecule. Protein molecules are unstable. So what we have developed a robust formulation in which the molecule is very stable. Now, there are two ways to do this clinical trial. One is that, you go through the Indian regulatory process, which is one of the most complex in the world. I don't think anybody was successful. We are the only one first successful, what is something called C3 approval. C3, C5, and it is a very long process. Maybe briefly, I will tell later. But we decided that we will go either to US or to Canada or to Mexico or to Australia or Netherlands. These are the regulatory bodies comparatively easy to get the approval. Now, there are two types of when you start the trial. One is called the phase 1 trial. The second possibility is that investigator supported trial. If you want to have a proof of concept. So we are focusing on two things. First, IST followed by investigational supported trial. Now, we will be, as a company, only will be able to do up to that level. Subsequent to phase 2, phase 3, worldwide clinical trials is very, very expensive. A phase 3 trial of this molecule will cost $400 to $500 million, which we can't do. On the other hand, up to this level, we have done up to about $12 to $15 million. The same way, diabetic retinopathy indications, again patent protected, high stability molecule, minimal toxicity. The IND filing will take some more time. Probably we are completing the whole process. These are our team. Professor Armugam is our chairman. Professor Satish is from US. He's one of our director. Professor Ambati, if you see Google search, you will see that he is one of the most famous ophthalmologists in the world. His brother, who got the other Ambati, he is. This is Jayakrishna Ambati. The other Ambati is the one who you can see in Guinness Book of World Record. He, at the age of 15, got MD from Harvard Medical School. So he is advising us regarding the clinical trials. So this is our lab, the people, and we have a complete facility for end-to-end development from beginning, whether it is cloning, expression, upstream, downstream formulation, and CGMP facility, but our animal experiments are done through collaborations. And that also everything we are doing in India. And one of the most complex thing, the entire process is, one is regulatory approval, the second is CGMP manufacturing, and of course, the approval for the phase 1 trial. Now, capabilities and accomplishments. Discovery was based on next generation phage display technology, which I will briefly tell. Demonstrated capabilities in discovery and development. Now, validated anti-VEGF biologics for 2 major indications. Fab 201 for the treatment of age-related macular degeneration, and we are ready for the phase 1, and the second one is for the diabetic retinopathy. Now, one of the major thing which I mentioned is that when you are developing a novel molecule, it has to be better than the existing molecule. If you are trying to develop another paracetamol with a different structure but similar capabilities, it will not be successful. They will not give the approval. So whenever you are making a novel molecule, whether it is biologics or NCE, you have to see what is the existing molecule. Either you have to go for a totally novel indication where there is nothing is there. For example, many of the cancers, it is possible. For example, anti-inflammatory we are developing, there is a series of anti-inflammatories there. So one of the molecules I was earlier developing were anti-inflammatory or anti-allergic molecule. So you require to develop a novel molecule which is far superior than the existing one. Otherwise, nobody will fund and you will not get the approval also. It is very important that, the patent protection should be there. If it is not patent protected, the value is 0. And not only that, the patent protection has to be worldwide. And high affinity and more efficacy. This is what we have focused. Can we get the molecule with high affinity? That means this antibody binds to the antigen, which is vascular endothelial growth factor, with the extreme high affinity. If extreme high affinity is there, naturally its efficacy will go up, dosage will come down, and probably because of low dose, the toxicity will not be there. Now, as I mentioned, for Wet-AMD, 12 injections are required. Can we reduce that? So another focus was, can it be made to 4 or 5 injections? The cost will come down. The other thing is that compliance will be there, affordability and availability. And this, if you really develop this in India, and the production cost is not going to be more than $4 or $5, and see it is sold for $2,000. So our idea is that, can we develop this molecule indigenously, and in India, can we introduce this molecule for, let's say, $10, $20 or $30 and internationally for maybe a higher cost? But definitely it will be competing with the existing molecule. Now, this is the product development path which we have done. Now, discovery phase is done using phage display technology. The classical methodology is that, once you have using PCR, amplify, and you can incorporate into the plasmid and transform into a bacterial system or a mammalian system or any of the cell system you can introduce. So that's the first part. Here what we have done is that synthetic biology and phage display technology. Now that itself will take an hour to explain, but I'll try to explain that briefly. The next part is that the molecular biology part, cloning of DNA sequence in the high yield expression vector. So it is very important. You need to have a very good expression vector. Then followed by upstream process. Upstream process means the fermentation process, followed by downstream process, bioanalytical, then in vivo studies. So these are the first set of experiments until the proof of concept is done. What is the proof of concept? In an animal model, you have to prove. Now, this is not a regulatory requirement, but if you are not doing that one, you yourself, the company itself will not know whether it's really going to be work. So first part is the proof of concept, followed by a series of upstream process, downstream process. We'll have to make sure this is a robust methodology, and multiple upstream process we have to do. For example, 5 repeat upstream process, downstream process, the formulation. This all has to be given to the regulatory people, and the data should be consistent. What are the major things? Fermentation process, purification process, development of the formulation, bioanalytical. There are two things are there. Analytical procedure, that means the quantifying, quality, then biochemical and biophysical characterization, like circular dichroism, then amino acid analysis, PI, SDS, so as SVP analysis. So what we are telling is that, a purified protein, absolutely nothing else, and non-configuration, non-conformation, and a robust testing methodology has to be there. Then this will be also finally tested for multiple tox study. But then after this will go for the preclinical studies, which is approved by government. You can't give it to anywhere, because this will have to be presented what all toxicity we are going to be done, and review committee on genetic manipulation will or the part of the DCGA, they will approve this. So multiple regulatory approval. These what we have mentioned, IBSC, multiple IBSC approvals, Institute Ethics Committee, then C1, C2, C3, C4, C5, then up to C11, C12. So these are, sorry, CT11, CT12, Form 29, Form 30. So just do not worry about this one. About 20 different approvals are required, governmental approvals, before starting the initiation of the material generation. That means the materials that is required for the clinical evaluation. Then from with this product, once you get the approval, you can go for the preclinical studies. Then CGMP manufacturing. CGMP means current good manufacturing practice. We can't do it in the lab. You have to have a separate place exclusively for this, and this is multiple approvals are there. Followed by IND filing. Now, as I mentioned, one is you can file in India, which will go through, but then it is very difficult to get an approval in India. So we are going to US. US comparatively easy, and there is a pre-IND approval process. That means you can also apply to US FDA an earlier stage and get their they will advise you how to go about, and multiple applications are there which we have done, and we have got now few queries. So in the next approval, we will get the approval to do either IST or clinical trial, phase 1 clinical trial. I assume that I have only 10 more minutes. So what I will do is that, I will very quickly go through. This is the process flow chart, what we are doing, the fab discovery part, then upstream processing part. These are all done in the lab. You can see what all different unit operations are there. Then downstream processing, each one has got like, about 5 or 6 unit operations are there. Then finally formulation also has 3 unit operations. Finally, the product is ready, and we will be also testing separately at every stage. Right from cloning onwards, we need to have the biochemical characterization has to be done. Mainly affinity, 2 determinations of affinity is there. So these are the processes. And what exactly is phage display? Phage is a virus which can infect the bacteria. Bacteriophage are viruses which infect bacterial cells. Enables expression of the foreign DNA purposely introduced in the vector in conjunction with a phage protein as a fusion protein for display on the phage surface. So if you see here, there is phage mid, like plasmid. In the phage mid will introduce various DNA sequences, which will represent an antibody structure or against VEGF. Now, which we need to have almost like 1 billion of such combinations of DNA sequences are there. And I don't know whether you are studying or not, students. It's a novel technology, probably 5 or 6 years. In India, few places it is there. But every novel biologics is coming via phage display technology. Then once this initial part is over, there is a panning process. That means VEGF will be immobilized, and we will see which all virus are getting attached to the VEGF molecule, and that will be taken. That means that will be having very high affinity. Now, this process will continue many times. We will end up getting 10, 15, 20 different virus which has got a high affinity antibody which can bind to VEGF. That is what the process is. Now, this is the structure of an anti-VEGF. This is the normal antibody, immunoglobulin G. You can see CDR, complementarity determining region. Now, this variable region and constant region consists of various, about both together about 12 CDRs will be there. This is constant region. What we are doing is only this part: variable region, light chain, variable region heavy chain, constant region light chain, constant region heavy chain. This is what we have developed, this part. And the most important thing is that you need to have a novel CDR. If we are using any of the CDR of the existing Lucentis, for example, existing competitors Lucentis, we won't get a patent and we can't develop that one. So it's very important, the novel CDR. Now, this is for the Wet-AMD indication. In India, there is, worldwide 30 million Wet-AMD patients worldwide. Probably about 5 million is there in India. Now, somebody who is suffering from Wet-AMD, this is, you can see, this is the normal vision, left side. Right hand side, the vision is like, impaired vision. So, and, after some time, you will completely lose the vision if you do not treat. That's why these drugs are used. So this is the clone development process, how we have cloned. Once we have the sequence, that has to be put into the plasmid. So how we have cloned Pvt Ltd? One is what is the gene details? Synthetic human anti-VEGF Fab molecule, bicistronic. If you see, I have shown you that Fab molecule which has got two chains. These two chains are independently cloned and put it into the same plasmid. Then after the protein is formed, the fusion takes place. Then vector information for a promoter. The promoter means, for A means the absence of phosphate, the induction takes place. It's a different type of induction process. Then PBR22 origin and carbapenem resistance, because we need to introduce carbapenem if unwanted bacteria should not come out. That's why. Then Pvt Ltd light chain 214 amino acid, heavy chain 228. Total is about 442 amino acids, and molecular weight is approximately 448 kilodalton. Now, this is a comparison what we have done in HUVEC assay. The human umbilical vascular endothelial cell lines. So that is what we have done. See, our Pvt Ltd is the most effective, and EC50 is the lowest compared to all other molecules. Exactly the same way we have done for the other molecules. Now, it is very important to develop a very high affinity molecule. That is number one. A stable molecule, no toxicity, highly efficacious, and number of injections has to be reduced. Now, when we presented at the regulatory people, what they wanted is that, they wanted to show us our CDRs are different. Now, it's a very complex experiment. Complementarity determining region. If our CDRs are same as, let's say, an existing molecule, we won't get patent, and the regulatory people, especially in the US and other places, they will reject, and we can go through a legal thing. So we had to prove that our what is paratope and epitope. So our paratopes are different. That means the place our amino acid CDR binds to VEGF by a different amino acid sequences. So that I can't explain this because it's a very complex experiment, but this is what we have done. Now, outline of upstream and downstream processing. So the first is the transformation, which I mentioned, then followed by clone screening, then master cell bank and working cell bank that normally is frozen. Then the process goes through, and cell biomass stored at -80 degree, and before that the fermentation. Normally, how the fermentation is done is that you might have about 100 ml, maybe 1 liter. Then it will go to a normal fermenter, 5 liter or 16 liter, 20 liter. We have range of fermenters are there. And please remember, fermenters are extremely expensive, and all the process can be controlled. So that is the upstream process. Then these are the downstream unit operations. First is cell lysis. Then we have developed a methodology in which, by heating up to a particular temperature, lot of unwanted proteins getting precipitated, and only some of the proteins of our interest remains. Almost 60-70% of the protein can be removed. So this is a very good process we have developed. And this this is one of the concentration method, TFF. Then 3 column purifications are there. Then finally you get the drug substance. So there are 2 products are there immediately after the purification called drug substance. But the drug substance has to be formulated immediately to get drug product. So the first what we are doing is the entire mechanism is for the development of drug substance, and subsequently immediately it is formulated, otherwise it will lose the stability. These are the various analytical assays what we have done. A quantitation assay by ELISA, in vitro binding assay, quantikind kit SPR, surface plasmon resonance, as well as the binding assay, in vitro efficacy assay using vascular endothelial growth factor induced STRET-RP1 proliferation assay, because this particular cell lines will not proliferate in the absence of VEGF. So then if our molecule goes and binds, you will be able to find out how much it is inhibited. Then protein product characterization, multiple tests are required to do this one, a large number of tests. Then in process related impurities. One of the major problem is that when you are using cell lines, host cell proteins will be there, host cell DNA will be there, and endotoxin will be there, and the leachate of the metrics what we are using. You have to make sure none of these things are present. Now the testing these in picomolar quantity is very complex. So one of the ways is that host cell DNA is that classical PCR method or RT-PCR method. Host cell protein we are measuring by developing an antibody in rabbit and testing that one. Endotoxin is the classical procedure. The same way protein L leachate also by ELISA method. Then product related impurities are tested by SDS, size exclusion, UPLC, RPHPLC, and SVP analysis. Then sterility assessment and confirmation. And one of the important thing when we went to US, what they have told is that whether your molecule is efficacious or not. I'm telling about the clinical trial principal investigator. The only thing they wanted to make sure that the molecule is absolutely sterile, because otherwise once it is injected into the eye can blow away. So that is one of the things. So there is a multiple process to make sure that what we are injecting into the eye is absolutely sterile. So this is the regulatory approval process. You can see the complexities in India. There is about 20-25 multiple approvals are there. Here is the major problem in anybody who is developing a molecule in India. But this much complexity is not there. And somebody who has worked on any pharmaceutical company, especially biological process, is amazingly complex. I do not want to explain too much on this. So what are the important key takeaways in our molecule? Our molecule is 2 times efficacy when compared to the existing molecule. You can see that one, the FAB201 compared to the existing molecule, and this is the vehicle. And another important thing what we have developed is that this molecule is extremely stable even at room temperature, which is normally not possible for any protein molecule. It gets denatured. So this is something we have done. And this is the final product what we have developed for injection, which will be taken to US under CGMP, what we have done. Now, the material generation has to be done in a CGMP place. Very complex procedure, very complex approval process. Normally, it takes months to get 1 gram, which has to be fill finish about 1,000 to 2,000 bottles. So this is the place where we are doing. We have the complete facilities there. Now, why formulation should be done? What is a formulation? The formulation consists of certain excipients which will stabilize the protein molecule. What's a drug product? Product is the formulated product. So we have developed this one, and the most important thing is that the glass vials used are sterile, depyrogenated, siliconized type 1 borosilicate glass, a very expensive glass. Otherwise, what will happen? Because we are using micrograms of protein that will bind to the sides of the silicon if it is a normal glass. So it's a very specialized this thing, and the stoppers also is very important. Now, please remember the regulatory people, every detail will have to give. And when they ask questions, we'll have to give the analytical details. Now, where we are going to do the clinical trials, as I mentioned, the first trial probably will be done in Mexico. Once you get the US FDA approval, you are allowed to do the trial either in USA or Mexico or in Canada. So that's what we are going to do. We have analyzed probably University of Kentucky, we are going to do the clinical trial. And only 5 patients will start with. That is called investigator supported trial. Comparatively, in 1 to 2 months time, we can complete. By doing this, our company's value suddenly goes up, provided the molecule do not have any adverse effect. It is going to be, if it is going to be a successful molecule. So after that only we will decide what is to be done. We will not be able to continue with the rest clinical trial because the cost involved is very high. Either VCs will further invest or we will sell the molecule. So the other is on diabetic retinopathy. Like, only 2 slides are there. A large number of people have got diabetic retinopathy, especially in India. So right now, only one molecule, the Lucentis, which is for Wet-AMD, is already approved for diabetic retinopathy. And we are developing, again, a novel molecule. Hopefully, the complete process will get into clinical trial in 6 months to a year's time. And we have formed a company in the US called Theragen Biologics Inc, because it is compulsory to have a company there so that it will be easy for the people to invest. And you can see that, the key features, and this is the clinical efficacy which we have done. And this is the no treatment, and after 5293 treatment, you can see very clearly it is effective. Now, this is the regulatory process what we have to go through in India, but then we have reasonably up to the clinical trial, we have got all the approvals in India. Now, whether we will do the clinical trials in India or not, we do not know at this point. So this is the last slide. Thank you very much. And I hope, in time I have finished. Thank you very much.